So mass transport does obviously include animals, but I'm going to particularly talk about today plants, because most students don't do very well when they talk about the good old plant.
That's nice, sir.
Right, let's get started. So, obviously mass transport in animals, you'll need to know about the heart, the lungs, gas exchange, cardiac cycle, all those kinds of things. But mass transport in plants, they particularly want you to know about the transport of waters and minerals in what vessel, please? The xylem, well done. So let's talk about the xylem first. So this slide is going to be about the xylem. Okay, so before we go on to talk about cohesion and tension theories, hello, let's talk about this. So first of all, the xylem tissue. So what does it transport? It transports water and mineral ions, okay, up the plant. Okay, they're very long, thin tubes, so they're long, continuous tubes. So they've got no end plates, continuous tubes. Any other features about xylem vessels, please? Yes, Rosie? They're dead, yeah, they're actually dead. They're joined end to end, dead cells with no end walls. So dead cells with no end walls. No end walls. Good. There we go. Now, this isn't in your spec, but I'm gonna say they're usually made of lignin, so they tend to be kind of like this. So these are the dead cells with no end walls, but they tend to have thickened walls to support, and that's made of lignin. I don't think that's in the spec, but there you go. Okay, and water. I haven't got a blue. Oh no. I have to use green there. So water and minerals are moving up. Right, okay, so what's amazing is that if you think of a tree, how tall is a tree? How tall is a fully grown tree? More than that, probably about 100 feet. So you've got to get water, gallons of water from the roots right up the stem, right up the trunk, into the leaves at the top of the tree. And you've got to do that throughout the day at large amounts of water and needed for photosynthesis and for turgidity of the cells. And you do that in complete silence. Isn't that amazing that it can do that? Oh, I'm sounding like David Attenborough now. Oh, my hero. Anyway, right, so, yeah, by the way, YouTubers, David Attenborough, Private Life of Plants, there's a really good thing about transpiration on there, so check that out. Right, okay. Right, so can anyone tell me what is the process called by which water moves up? Yes. Well, transpiration drives it, but we're going to talk about now. We're going to talk about a process called the cohesion tension theory. Cohesion tension theory, and within that is respiration. Okay. It is a theory. Okay. So the first thing to state is, if you think about a leaf, okay, so at the top of the leaf is my leaf. going on to a branch, onto a stem. Okay, so on the bottom here, I've got my, what have I got lots of on the bottom of the leaf? Yeah, I've got my stomata, yeah. So stomata can control the gas exchange, so controlling exchange of gases, open and close. I won't go into that now. And obviously, a kind of, I'm going to use black as, right, that's going to be a water molecule. Okay. So water can move through, and it obviously moves into some of the cells. What's it going to be used for? Sorry? Used for photosynthesis and also to maintain turgidity. Okay. Turgid cells, okay. And it's kind of a byproduct of that is evaporation. So the water molecules are going to be taken out through diffusion, okay. So we're going to diffuse out of the leaf, okay. Now, anyone tell me some features about water molecules? What do you know about H2O? So water molecules, tell me something about water. Do you know this relates to paper, other stuff in paper one? Yeah. Good, polar. So you've got hydrogen bonds, well done. Yeah. What else? They're cohesive, basically. They're cohesive molecules. They tend to stick together. Cohesive, so they bind together easily. Okay, and that's an important feature of water when you're talking about cohesion tension theory. Right, so cohesion tension, basically one, evaporation through the stomata, evaporation of water through stomata. Okay, so what will happen to the water column is this creates tension, okay? So this creates tension or suction, which pulls more water into the leaf. So that creates tension. As the water molecules are being replaced. In the leaf. So this means that the water column will then move upwards through the stem, okay, because the water is cohesive. The whole column of water will move upwards. The whole column moves up due to as they are cohesive. The water molecules are cohesive. And thus, all creates a suction pressure. Its pressure changes. So it moves upwards. Right, okay. So can anyone remember how we measure transpiration? We use the transpiration is the evaporation of water at the leaf. So, yep. It is the potometer. Good. So potometer. So potometer is a, I'm not going to go through it in a lot of detail. So basically it's a sealed system. So it's sealed. You can measure distance and the time, the distance of water over time. In a capillary tube. And guess what they love to ask you? they love to ask about the rates of transpiration. So there's usually are calculations related to that. Now, I suggest you find some questions on pedometers. Okay, so if you go to physics and maths tutor, there are other sites as well, and you can find, if you look up transpiration, mass transport, you should be able to find some pedometer questions. I really suggest you do that strongly. Also, we need to know about what affects transpiration rates. So transpiration rates are affected by what? What kind of things affect the rates of transpiration in a plant? Yes, Rosie. So an increase in temp, so you get more evaporation. Well done. Good. Any others? Yes, Elliot. No, I wouldn't. Well, yes, but I would not say it's about affecting transpiration rates. Yeah. Humidity, very good. So humidity is a measure of the amount of water vapor in the air. So humidity, if it's high, okay, that reduces transpiration because there's a lower concentration gradient between the inside and outside. So it lowers the concentration gradient between the inside and outside of the leaf. Okay. Humidity. And then things like light, obviously, and the amount of wind as well. So you talk about light intensity, and obviously wind speed as well. And what the examiners might do is link with what? What could they link this with? Possibly. How about sorry? No, they would link it to adaptations. The adaptation, oh goodness me, adaptations in plants. So things like xerophytes. Have you heard of xerophytes? Yep. So xerophytic adaptations to living in conditions where there's very little water. So sunken stomata, rolled leaves, hairs, less stomata, those kinds of things. We were supposed to go on a field trip, but due to COVID, unfortunately, DrBiology could not take his amazing students away. Right, thank you. Right, so pedometers, good. Let's talk about Phloem then. Right. Phloem. It is a shame we haven't gone, because I could have used some of those photos in the leavers assembly next week. Yeah. Right, let's talk about Phloem. So first of all, what does it transport? Mainly? Yeah. What is it? So mainly sugars like sucrose, so transport solutes. The best thing to do is transport solutes, and that tends to be sucrose. Okay. Like sucrose. Okay, now the phloem is a bit more interesting because it is living cells, okay? And in terms of the transport, you've got to think about the phloem. First of all, well, if they're living cells, so the first thing to talk about is sieve tube elements. Okay, they have very few organelles in that cell. So next to it, so if that's the, let's say, I don't know, let's say this is the, oh, there's some end walls with holes in them as well. So that's the sieve tube. But next to the sieve tube, what's next to the sieve tube, guys? Does anyone remember? Companion cells, good. So these companion cells do have, they have lots of features in them, okay? So their nucleus, their vacuole, etc. So companion cells. And they carry out the functions, so they actually have lots of mitochondria as well. So there you go. If you ever want to talk about mitochondria, you can talk about companion cells. Why? Because there's a lot of active transport going on. In fact, it's a telltale sign. If there's lots of mitochondria, then think active transport. We'll talk about it now. Okay, so the movement of solutes, what do we actually call that? What's the process by which solutes move up and down the plant? Can you remember? It's not transpiration, it sounds like transpiration. Yeah, well done. It's called translocation. And basically, there's two other kind of things you need to think about. You need to think about things that we call sources. So source, okay, to sink. And we'll talk about sources to sinks. Now, so source is where there is lots of solutes, lots of sugars, and the sink is where there's very few sugars. Okay, that's where it's moving to. So usually the source could be, if it's in the summer, it would be in the leaf, basically. Whereas in the spring, it will be more likely in the roots. Okay. But moves things from sources to sinks. Right. So this brings us neatly to the mass flow hypothesis. What do I mean by a hypothesis? What do I mean by a hypothesis? It's not certain, yeah, it's theory basically. So scientists have tested it, but they don't exactly know how they're transported. Okay, this is a supported theory, but it's still a theory. Okay, so first of all, we need to talk about the source end. Okay, so let's talk about the source end. So first of all, number one, we're at the source. So that usually is, for example, e.g. at sites of photosynthesis. Photosynthesis, hopefully they're not talking about me outside. No, I'm pretty sure they're not. It's good. Okay, so they active transport means that I'm going to call it AT. It actively loads sugars into the phloem, into the sieve tubes. And what should I call it? Into the sieve tubes from the companion cells. Okay, so into sieve tubes. All right, so it's actively pushing it in. Right. So I'm just going to draw the sieve tube here. Okay. So here's the sieve tube, and I'm actively putting loads and loads and loads and loads of sugar, sucrose, into this part of the thing. Okay. What have I done? If that goes in, what am I doing to the inside of the sieve tube in terms of the water potential? I'm decreasing it, yeah, there's a lower water potential. Now, if there's a lower water potential, okay, that means that water will move in because right next to it, I haven't explained this to you, but pretty near to it is xylem, okay? So here's the xylem, all right? And some of the water, there's lots of water in the xylem that will move into there. So lower water potential, so water moves in, okay? All right, so lots of sucrose, lots of water in the source. So what will that create? Pressure, so it creates a higher pressure, a high pressure, wherever it might be. It could be at the bottom of the plant, at the top of the plant, but it creates a high pressure. And as we know, in pressure changes, you know, like with the heart and blood and everything else, okay, it will move, right? But it won't move because now we need to think about, that's the source, that's what's happening at the source end, what's happening at the sink end. So at the sink, the place it's going to move to, solutes are being removed from the phloem. So solutes removed from phloem. To be used, okay? So they might be used up. So here's my sink here. So solutes are being taken out. Okay. First of all, through the companion cells. And here's my xylem again. So if I have a higher water potential, okay, I have a higher water potential. And in the xylem, what will happen to the water? Yeah, so by osmosis, it will move back out, and actually it doesn't all go into the xylem, some will go into the cells as well. So water moves out. Okay, so if you're getting solutes and water moving out, this lowers the pressure. So this creates a lower pressure in the sink. Okay. Oh yeah, I forgot to tell you. What do you think the solutes are going to be used for? The sugars. Yeah, good. So we need to say that it can be used for respiration. Very good. Also combining with nutrients, nitrates to make proteins, but there we go. So what happens? This results finally, can you see that in the picture? Results in a lower, this results, sorry, in a pressure gradient from the source to the sink. And you can see why it's a good hypothesis, because it kind of makes sense. Right. One of the key things, I haven't quite finished, there's a couple of other things. I've got about 2 minutes to go through this. More than likely, okay, they will talk about experiments. So you might have to evaluate evidence, okay, and the key bits of evidence is things like removing bark. So what they might do is show you a piece of bark, and they've kind of, what they've done is they've kind of cut the phloem, okay? So when they cut the phloem, it affects the growth of the bark. Okay. Also, radioactive tracers. So they might talk about tracers. So 14 carbon. So use of 14 carbon to see the movement of the sugars. Okay. Movement of organic substances. What else can feed off the phloem? If this was the plant, what animal can feed off it? My favourite animal, by the way. Aphids. Aphids. There you go. So aphids have stylets like that, and they actually feed off the phloem. Oh God, it's not very good aphid, but there you go. So they might be able to measure the pressure, for example. So they could measure the pressure gradient. So there we go. Again, it's well worth looking at some applied questions on that in terms of transport through the phloem. Thanks for telling the world that, Elliot. Yeah. See you soon. Half an hour. Oh yes, I'm teaching in half an hour. Right. Okay. Right. Any questions on that? Is that quite useful just to quickly go through it all? Do check out some exam questions, though. They do like to ask quite applied questions on this. All right. So thank you very much, guys. Thank you, everybody, for coming today, and I'll see you soon.